Hello and welcome. Thank you so much for joining us on the Inside Track. I'm Jennifer Strong, the editor for audio and live journalism at Tech Review, also host of a new podcast called In Machines We Trust. So I'm here this week with a behind-the-scenes look at MIT's labs. We kicked it off yesterday with the nuclear reactor, also known as MITR. Today we're going to be going inside the Broad Institute. Here to be our tour guide for this one is Stacey Gabriel. She's senior director of the genomics platform there, which under her guidance operates as one of the largest sequencing centers in the world. Welcome, Stacey.
Thanks, Jennifer. I'm happy to be here today.
So you made a video tour, which you're going to show us. It is simply a delight. I don't know if you've ever been a Mr. Rogers fan, but you know that moment as a kid when you get to see how crayons are made is something I've never forgotten. So anyway, for those of us who love labs, this is a similar kind of pulling back the curtain.
Great. Yeah, I'm excited to show you all. And it's true, we usually describe ourselves just as one of the largest DNA sequencing centers in the world. But what the tour is going to show you today, and the tour is hosted by some of my team, we're going to show you how we've turned that lab into a very large COVID diagnostic lab.
All right, folks, she's going to stick around after the video to answer your questions, and you should feel free to start entering them into the chat just anytime. With that, let's go have a look.
My name is Sheila Dodge, and I'm the general manager of the genomics group here at the Broad Institute, where we're doing COVID testing diagnostics 24/7. So when this all first started, we were basically a sequencing laboratory. So these were the only two machines that would detect COVID virus that we had. And this is really where it all started. And so within a very short period, roughly 2 weeks, we were able to stand up a clinical diagnostic test for COVID-19 to generate hundreds of samples a day, and eventually we got to 1,000 a day. But we knew that wasn't enough. We thought, let's redesign this process to make it really highly scalable. So we moved from these first 2 machines to then expanding our capacity to a very large number of machines that could do hundreds of thousands of samples a day on these machines. And this allows us to enable testing in underserved populations, in colleges and universities, in nursing homes, skilled nursing homes, in all different types of settings that allow people to get really fast turnaround COVID testing done, and enables us to find and contain outbreaks fast before they get too big. This lab is where we actually send out kits. And so this is where all of the swabs and the tubes and the necessary materials for collection sites come together. We're sending to hundreds of groups a day. Hundreds of thousands of tubes and swabs go out every day. And those sites get everything they need to do the COVID testing from us. We allow sample drop-offs 24 hours a day, 7 days a week, right through these front doors. We have couriers coming from all over New England that are dropping off samples. So these samples will come upstairs, go through these doors here, and into the COVID testing lab. The first step here is receipt and accessioning. To talk a little bit about kind of what this process is and to get those tubes out of the boxes and ready to go into the lab, we have Emily here with us today. Emily is one of our general supervisors that helps manage this part of the lab here.
As soon as samples come in the door, we do a intake and receipt process on them, which is basically making sure that the samples came from a site that we expected, that they weren't damaged in transit, that the samples didn't spill out of their boxes when they were coming into us, to make sure that all the samples were transited across the country or between states in a safe way. The next step of the process is doing a inspection of the package, where they count all the samples that were in the box to make sure that the number of samples in that shipment matches the manifest that we have. And then they go through and then methodically double check all of the PHI, including the patient's name, date of birth, and the sample ID numbers on each of the tubes. And then once they've confirmed each of the samples, they'll go into one of two areas. They'll either go into one of these blue racks, which indicates that the sample has gone through the accessioning process, or they'll be placed into a black Hamilton carrier. And that is one of the pieces that we use in the next downstream part of the process. So a third place that the sample can end up is in one of these orange racks, which indicates that there's been some sort of problem with the sample, or that it wasn't found informatically in our system, and that we need to get in touch with a PM or a site in order to get some more information to get the sample ready to go downstream. Once these samples have been loaded into the Hamilton carriers, they get put onto these carts that get put into another part of our first in, first out pull system. So once a cart is filled, we move it to the front of this pull section, which is this green tape along the floor, and this cart is ready to go downstream in the process.
So the next part of the process is for us to take the tubes into the first automated section of the lab. And in this section here, we have Tammy Mason, who's actually the manager of the entire laboratory process here for the COVID-19 testing, that's going to take us through what happens to those tubes once they've been accessioned.
So the first step after accessioning is decapping. So we've implemented a high-scale decapping machine. It decaps at a rate of 1,200 samples per hour, and it is continuously filled with more samples. And as they are uncapped, they go on to the next workflow. This is our tube filler. This tube filler is filling a set of 8 carriers, 188 samples, with a lysis buffer that inactivates the virus. Once this buffer is added, the samples are now inert and are much more safe to use. So once the samples are filled with the lysis buffer, they go onto the shaker. This shaker will run for 15 minutes. And what this is doing is taking the cells from the swab into the liquid. So the tubes are transferred from the shakers onto these Hamilton Starlets. The Hamilton Starlet is an automated robot. Its only function is to transfer liquid from one place to another, and we use it at a very high capacity in order to transfer the liquid from the sample swab tubes into a 96-well plate, which is something that we can use much more easily throughout our process. Once the samples are complete on the Hamilton, they output a plate. This plate now contains 94 samples and 2 controls, and it's barcoded so that we can track this plate through the lab.
Once the plates are now in this format, they're ready for the next step of the process, which is, as we move through, to extracting the RNA material. So I'm going to have Brendan, who's actually been the leader that's developed a lot of the process that you've seen here today, talk us through kind of what the next steps of this are and take us into the extraction and qPCR.
Okay, so throughout the process that you've seen so far, the samples have been reduced in their footprint more and more and more. And what came out of transfers are these 96 well plates holding 96 actual clinical samples. At this step, 4 of those 96 well plates are now being consolidated further into a single 384 well plate. It's in that plate that the actual RNA will be extracted from the sample. So one of the things to point out is that there's a lot of different methods for actually extracting RNA from a clinical sample. And when we designed this process, we realized that we needed a method that was very automatable. We settled on this one particular kit, and the way it works is the RNA is bound to these small magnetic beads. The beads are then pulled down onto a magnet, and the residual cellular material or sample from the swab is washed away. A couple subsequent washes then clean that RNA further, and then that clean RNA is eluted off those beads into the sample that will then take onto qPCR. Okay, so at this step, that clean extracted RNA is now being transferred into a plate that already contains the master mix for the RT qPCR reaction. It's at this step that everything's ready to go onto our detection systems that will then actually detect the signal as to whether some individual has COVID or not.
Then we move them to the detection instruments, which are out around the corner. And so here in the detection lab is where we're, looking for whether or not each sample actually has viral molecules in it. And so these machines here are detection machines. These are QuantStudios that allow us to look in real time at when certain molecules start to amplify in these particular samples.
If a clinical sample has COVID present, fragments of that RNA are transcribed into DNA. And then as this machine heats and cools cycle after cycle, it doubles every cycle the number of fragments of the COVID transcribed DNA. If somebody has a lot of copies and has a high viral load, you'll get a signal pretty early in the process. But because this test is so sensitive, even a sample that has a handful of copies, maybe 50 copies, you'll end up getting signal, but it'll be later in the process. And that's what this machine is doing. Cycle after cycle, it's measuring the intensity of the fluorescence of that individual sample. And then what it does is it plots it on this curve. And by looking at the curve, you can actually see whether somebody had amplifiable COVID RNA on their swab.
And so what happens after this is once we've reviewed this data and we're confident that we know that the sample is positive or negative, then we have our resulting software sort of pick that information up, we process it, and then we result back out to all the sites. So whether that's directly to the participants themselves, which is most of the sites, or to their physician or the ordering physician that actually ordered the test from us and sent the tubes to us.
Welcome back. I'm Jennifer Strong from Tech Review, and I'm here with Stacey Gabriel. She's Senior Director of the Genomics Platform at the Broad. Folks, the floor is open, so please send in your questions. And Stacey, while those come in, every one of us has made big adjustments because of COVID, right? I mean, just look at where we are right now. But this was a huge pivot for you at the lab, simply huge. Can you tell us what that was like?
It was an enormous pivot, yes, and it happened very quickly, as you can imagine. It happened over, as we said in the video, a matter of really days of, one of our core faculty members, who's also an infectious disease doctor, calling me up, on March 8th, I remember the day very clearly, saying to me, Stacey, testing is scaling so poorly. What can you do about it? You've got a CLIA lab, you've got all this automation, you've got all these people who understand large scale activities. What can you do? And so I said, what's the test? I didn't know what the test was. And so we figured it out. It was a PCR test. And on March 23rd, we had a validated assay and submitted our application to the FDA for emergency use authorization. So it was extremely quick that we pulled this together.
Wow. We all live and learn with anything we do, right? I'm wondering here, what are some adjustments you've made at the lab now based on what you found to be the best practices here?
Yeah, so many things. We've grown massively in size. So I guess the first thing I'll say is, our team was about 120 people doing our DNA sequencing activity. We've hired 250 people since May.
Wow.
Yes. So we had to scale up. We've created all kinds of new types of positions we never had before. People who do that we showed you that activity of accessioning the tube. So you're bringing the tube in, you're barcode scanning it, making sure the information is correct. So on each of our shifts, and we do operate 24/7, on each of our shifts, we have 42 people just doing that activity, and that will likely scale a little bit from there. So we've learned that. We had to build entirely new informatics systems. Even though we had been operating a clinical lab, we weren't receiving diagnostic test orders in this fashion, right? So we had to create an entirely new, and we actually outsourced to not outsourced, but we insourced a third-party software to help us do that. An existing company, someone that was already supporting ordering and resulting, a company called LK actually provides our software. So that had to be created, and those are two of the pretty big things, I would say. Just the massive staffing lift, the informatics and the ordering piece, and then also just figuring out the whole thing about how we're going to actually scale. And we changed a lot about how samples come in, and we can talk more about that if there's interest, but I want to let people ask questions as needed.
Well, to that end, Mac is asking, what are the top 3 things you and your team have learned from this?
Be fearless. Don't listen to the hype. I think that early in those days of scaling and COVID testing, all you heard about was supply chain. Right. Oh, you can't get this, you can't get that. And so, we just really we're pretty determined to figure our ways around that. I would say that's number 1. I'd say number 2 is the, importance and the diversity of types of roles and thinking about labs in certain ways. So as you can imagine, this is not your traditional diagnostic or clinical lab. Like, we run this very much like a factory, with the same sort of like industrial principles that you would see applied at Toyota or Harley Davidson or whatever. And so, and we're very fortunate that our folks are expert in sort of like this industrial process design. So I would say that was an enormous thing. And the third thing I would say is just the, importance of really good partners in learning, good hospital partners, good users on the back end, all of this.
Well, Heidi is asking, are you planning on keeping this COVID testing going through the duration of the pandemic? And what happened to the work you were doing before all of this? Like, has that been paused or moved somewhere else?
Yes. So, we're going to test as long as it's needed. It turns out, we're pretty good at it. And so maybe we'll figure out other tests to do. But we're going to keep doing this test absolutely as long as it's needed. We are back to our normal work, believe it or not. So our sequencing activity resumed. The Broad itself went through a bit of a ramp down, through those months of April to end of May, and we ramped down sequencing a lot then. But we are scaling back up our sequencing operations. We're a big part of a national sort of biobank project called the All of Us Initiative, funded by the NIH, and that project's going. We're sequencing now for that, every day. So we're very busy, and the machines are running, the buildings are full, but we're back at it.
Yeah, I would say I've been in this building. It is not as large as you might think for all the things you're doing. Steve is asking, how is the COVID testing presumably being funded?
Yeah, great question. So the Broad Institute, as you may know, is a nonprofit research institute. So we're selling this test, so we're charging people for the test who are ordering it from us, but it's not being done at a profit. Basically, we've priced it at our cost, essentially, and that price ranges between $25 and $35, and depending on the program and the volume people are ordering in. We have received some funding for some infrastructure development and some of the early startup. from some philanthropic sources, from the NIH, through this RADx program you may have heard of, and from the state of Massachusetts as well.
What's your biggest challenge right now?
Our biggest challenge right now is, well, I think it's trying to have a crystal ball, I guess, and trying to figure out, like what is going to happen with, these numbers we're seeing throughout the country are ticking up. And so really trying to figure out how we can be most responsive, and are we going to have to expand a lot? Is it going to slow down because some magic new, 3-minute test is going to come out? So right. We're sort of always on this edge of wanting to provide the right thing at the right time, and really trying to keep our eye on it. So that's a big challenge.
Sure. Is there something that you wish people better understood about your work right now?
That's a good question. I think people have, a misunderstanding of like everything it takes to get these tests sort of working, at scale. Again, it's not just the molecular biology, it's sort of the whole, infrastructure around the activity itself. I mean, there's all kinds of things that we learned. I mean, keep in mind, this is still a diagnostic test. It has to be ordered by a physician. Results are returned in a way that, the physician, particularly if a result is positive, should return that result. And so, we do get a lot of interest from all kinds of communities across the country of, can you test my high school? Can you test my kindergarten? Can you test, and it's like, I think that there needs to be a, I guess more of a strategy around how that testing would be efficiently ordered and returned, and how those results would be used, right, to manage outbreaks, to manage, bringing people back to school, back to work. So I think that's, probably a little bit of what is, I would say lacking the most.
For sure. Now, I think it might be really interesting if we just took a giant step back for a second, because the Broad has been doing cutting edge genomics work for decades before, COVID got here.
Yeah.
So maybe give us a little bit of a history lesson.
Oh, sure. Yeah, right. So our lab, our history goes back to the Human Genome Project. So about 30% of the first human genome was sequenced in the same building you just saw a tour of. And again, all along, there's been a really big focus of, the industrial operations and thinking at scale. And so we applied that from the sequencing the first human genome to now, tens of thousands of human genomes, and we'll be at millions of human genomes before you know it. And so we're really committed to that sort of driving the technology forward, scaling, and really enabling the next big scientific question.
So we have another audience question out here, and they're asking, are you testing samples collected using saliva, blood, a variety, any specific biosample challenges?
Sure. So for the diagnostic test, for the large scale activity, we transitioned solely to the anterior nasal swabs. So these are the little swabs that are like Q-tips that you just kind of run around the front of your nose. It's not the more sort of invasive, NP swab. And we completely standard on one type of swab, one type of tube. The tube is received dry. There's no media in it because we had a history of leaky tubes for a few months that were terrible. So that was one of our biosample challenges. We stayed away from the saliva. We didn't, the saliva didn't work as well with our automation because, as we tried to put across in the video, every step of that process is basically handled by a liquid handling robot. And so what we were just experiencing was that saliva was just a little bit more variable in terms of viscosity. It wasn't really worth it for us. And the collection devices were more expensive. Like right now, our swab and tube costs like $1, I think, combined. And they're made at massive quantities, because they're just these, our tube is a Vacutainer tube. We buy it from BD. It's the kind of tube that, hospitals use for blood collection. So they're made in massive quantity. So, I think we learned a lot, again, in those early days of all the constraints around NP swabs and VTM, and really tried to, develop a system that was really robust to supply chain challenges. Not to say they don't come up, but they do.
Sure. So Iker is asking from the audience whether you're sharing what you're learning here and helping other facilities around the world to establish and improve their capacities.
Yeah, we're certainly doing our best. There was an article you may have seen in the Wall Street Journal a couple of weeks ago. A group in Austin, Texas, UT Southwestern, adopted basically we call the system the pod, with the idea that a pod could do 100,000 tests a day, and then inside the pod are podlets of 25,000. So 4 podlets make a pod. So they adopted that idea and are, they're scaling to tens of thousands per day. There's a group at GW in DC, I think, that they're trying to take this on. We're trying our best to get it out there. I mean, people continually ask us to write up a paper about it, but honestly,
in all your free time,
we haven't had time with all of the work with the FDA and getting our EUAs through and running the lab, but we will get it out there. I think it's really important for others to see what's possible.
Sure. Now we're starting to run out of time, but quickly, have there been any big surprises?
Big surprises. Everything's been a surprise. I mean, I would say the surprise to me is how in demand this still continues to be today.
Yeah.
Right. I mean, we're 7 months into this right now. And every day we get inquiries that are from across the country, across the world, of people really wanting testing capacity. So every day I'm surprised by that there's not, more of I would say, a national coordinated response. But yeah, it just doesn't slow down.
Well, with that, it's time for me to send folks back into the conference. That's Stacey Gabriel of the Broad Institute's Genomics platform. Thank you so much for taking the time to chat.
Great. Thanks for the interest. Thanks very much. Enjoy the show.
Thanks, folks. I hope we see you back here again tomorrow when we go visit MIT Nano. Until then, take care.
Great. Bye.